All right, let's talk about one of the coolest systems in your body, the MHC Class 1 molecule. You can think of it like this super sophisticated security system that's always on patrol, looking for bad guys hiding in the one place you'd never think to look, inside your own cells. So, let's get right into it.
Okay, so here's how we're going to break this down. First, we'll look at the problem, this whole inside job thing. Then we'll check out the solution, the cellular ID card. After that, we'll meet the security patrol that's checking those IDs. And finally, we'll wrap up with why this matters so much to you and your health.
All right, so first things first, let's talk about the big challenge your immune system is up against. See, it's not always about fighting enemies that are, floating around in your blood. The real tricky part is finding the invaders that have already gotten inside, the ones that have breached the walls of your cells.
And that brings us to the big question, right? How in the world does your body find a virus or even a cancer cell when it's hiding inside one of your own cells? Think about it. A virus's whole game plan is to sneak inside a cell and hijack its machinery to make more of itself. So how do you spot a cell that looks perfectly fine from the outside, but is secretly a virus factory on the inside?
Yeah, that's really the heart of the problem. It's a classic inside job. The enemy isn't some outsider anymore. It's taken over from within. Immunologists have a fancy term for this, endogenous antigens, but it just means the threat is coming from the inside, and that makes finding it super tricky. You can't just look for something foreign, because that something is now part of the cell itself.
So to deal with this inside job dilemma, the body came up with an absolutely brilliant solution. It basically forces every single cell to constantly show its work, to report on exactly what's going on inside. And that report? Well, that's where the MHC Class 1 molecule comes in.
Now I know, major histocompatibility complex, class 1. It's a mouthful, but the idea behind it is actually really simple. Just picture the MHC1 molecule as a tiny little display stand on the outside of the cell. And what's it showing off? It's displaying little pieces, or peptides, of all the proteins that are being made inside that cell at any given moment.
Honestly, the best way to think about it is like a universal ID card. Every single cell has to constantly hold up this ID that basically says, hey, here's a sample of what I'm currently building. Now, if the cell's healthy, that ID just shows normal, everyday self-proteins, no problem. But if it's infected with a virus, that ID card is going to show something foreign, a dead giveaway that something is wrong. And what's amazing is that this security check is happening everywhere. I mean, everywhere. Pretty much every single cell in your body that has a nucleus, and that's almost all of them, has these MHC1 molecules on its surface. The main exception? Red blood cells. They don't have a nucleus, so they don't have the gear to do this.
So you might be wondering, how does this ID card get made? Well, it's like a non-stop assembly line. First, proteins inside the cell get chopped up into tiny little pieces called peptides. Think of it like a paper shredder. Then, those little peptide pieces get shipped over to a part of the cell called the endoplasmic reticulum, or ER. Inside the ER, one of those peptides gets loaded on a fresh MHC1 molecule. And finally, that whole package travels to the cell surface and puts itself on display for any passing immune cells to see.
And here we can see a picture of that whole process. You can see the protein bits getting made, heading into the ER, and then getting attached to the MHC1 molecule right before it goes to the surface. It looks complicated for sure, but the end result is super simple. The cell is being totally transparent about what's happening on the inside.
And get this whole system is unbelievably fast. We're not talking days here. In fact, within about an hour and a half of a flu virus infecting a cell, that cell is already showing pieces of the virus on its surface. It's basically screaming for help, waving this giant red flag. That is an incredibly fast response.
Okay, so you've got all these cells showing their ID cards, but an ID is useless if no one's checking it, right? Exactly. And that brings us to the next part of the story, the security patrol. The specialized immune cells whose whole job is to go around and check these IDs.
And these security guards have a name. They're called cytotoxic T cells. You might also hear them called CD8 positive T cells. Their mission, should they choose to accept it, is to patrol all through your body, constantly scanning the MHC1 molecules on every single cell they bump into, just looking for anything suspicious. So this T cell is basically trained to spot the difference between friend and foe. If it checks a cell's ID and sees a normal self-peptide, just a regular piece of a human protein, it's like, okay, you're good. It gives the all clear and just moves on. But if it inspects an ID card and sees a foreign peptide, maybe from a virus or a weird mutated one from a cancer cell, beep, 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 the alarms go off, threat detected.
And this check isn't just a quick glance. It's a full-on physical inspection. The T cell has this special receptor that literally fits over the MHCI molecule, like a key in a lock. This allows it to read exactly what peptide is being displayed. It's this super specific molecular handshake to verify identity.
And this is the critical moment, when that T cell's receptor locks on to an MHCI that's holding a foreign peptide. That's the trigger. That's signal number 1 for the T cell to activate. The security guard has found an intruder. It's go time.
And when these T cells act, they don't mess around. The response is swift and it's lethal. The T cell doesn't try to save the infected cell. It's too late for that. It eliminates the threat. It does this by releasing 2 key proteins. First, perforins, which, just like they sound, perforate the cell. They punch holes in its outer membrane. Then, granzymes rush in through those holes and trigger a self-destruct program called apoptosis. It's basically a clean, controlled demolition to take the compromised cell off the board.
Okay, for our last section, let's zoom out a little bit. Why does all this matter? Why is understanding this system so important for, medicine and just our everyday health?
A really good way to lock in what MHCI does is to compare it to its sibling, MHC2. They're related, but they are very different jobs. So MHC1, the one we've been talking about, deals with internal threats, think viruses and cancer, and it talks to the killer T cells. MHC2, on the other hand, deals with external threats, like bacteria that have been eaten up. And it doesn't talk to killer T cells, it talks to helper T cells, the ones that coordinate the whole immune response.
And the importance of this system? Wow, it's hard to overstate. It's our #1 defense against viruses like the flu or HIV. It's our body's 24/7 cancer surveillance program, letting T cells hunt down and destroy mutated cells. It's also the reason why organ transplants need such careful tissue matching. You have to make sure the recipient's T cells don't see the new organ as a foreign threat. And of course, this whole pathway is a major focus for developing new and better vaccines.
So that just leaves us with one big final question. If this system is so good, how do some viruses and cancers actually get away? Well, it turns out this system isn't foolproof. It's a constant arms race. And some of the most successful pathogens and cancers have evolved some seriously clever ways to mess with this system, to basically hide their ID cards from the T cell patrol. And figuring out how they do that, that's one of the most exciting frontiers in immunology right now.